In Unit 4 Lab 3, what we're going to do is we're going to look at the process of gametogenesis, the formation of sperm and eggs, and meiosis, and then relate that to structures in the ovary and the testes by looking at models and under a microscope. And so we won't do the microscope stuff on the video, but we'll do the other. So just as a quick review of meiosis, the role of meiosis is to reduce chromosome numbers from 46 in human cells to 23. So we divide meiosis into two divisions. So at the top here is meiosis 1, and this is metaphase of meiosis 1, where homologous pairs line up. So cells that are undergoing meiosis are called primary cells before meiosis 1. Once a cell has undergone meiosis 1, as we see here in Anaphase, and we get the two daughter cells, then the daughter cells formed from meiosis 1 are called secondary cells, and then the end product of meiosis 2 is a gamete in gametogenesis. So this is a classic model for sperm production. And so the cells that would be getting ready for meiosis 1 division would be called primary spermatocytes. The end product of meiosis 1 would be 2 secondary spermatocytes. And then the end product of meiosis 2 would be 4 cells that are called spermatotids. Now, if we were relating that to an ovary, then the same pattern is true in that the cells that would be before meiosis 1 are primary oocytes. The product of meiosis 1 would be a secondary oocyte. The difference is that because the cytoplasm is critical for early development, that if this was an example of oogenesis, one cell would retain all the cytoplasm, and the second nucleus would be injected as a polar body, which we'll talk about later. And then the end product of meiosis 2 would be the ovum, and we'd have one cell with all the cytoplasm and 2 polar bodies. So it's very different for that reason. So what we're going to look at first is how these cells, primary oocytes and secondary oocytes, relate to what we would see in an ovary. So this board represents the formation of an oocyte or ovum within an ovary. So the area in which cells, primary oocytes, are housed are called follicles. So everything we see over here that's pink is a progression of follicles that takes place. So when we think of an ovarian cycle, the classic ovarian cycle is 28 days long, and the ovarian cycle starts when a woman has her first day of bleeding on her period, and that would be day 1. So during day 1, all of the eggs in the ovaries are like this, and they're housed in a single row of cells that surround them that's called a follicle. And this follicle is called a primordial follicle. During embryonic development, all the primordial follicles in the ovary were formed during the formation of the ovary. And then nothing else goes on until a woman starts having her periods. Girls go through puberty and have their periods. And then they begin to develop 1 egg per month. So at day 1, we have a primordial follicle, and a hormone that we learned from the pituitary gland, follicle stimulating hormone, begins to increase in production, and it targets these follicles in the ovary. So as follicle stimulating hormone increases, the follicular cells around the ovary begin to be, around the follicle, in the ovary, begin to be active. And we can see that these are very thin cells, and now the cells have begun to increase in size. and the cells will eventually undergo mitosis. So as soon as we get more than one row of cells, like in this example, we no longer have a primordial follicle, which these two are, but we have a primary follicle. And in a primordial follicle and in a primary follicle, we still have a primary oocyte, a cell that has not undergone meiosis 1 yet. So we have an early primary follicle here, kind of a middle primary follicle, and a late primary follicle. As you're looking at this board, they're trying to show you that the size of the follicle is increasing, and to do that they're making the primary oocyte look smaller. But the primary oocyte's not changing in size, the follicle's actually getting larger. All right. Now what happens as follicle stimulating hormone continues is the follicle continues to develop, and it'll develop a space or a cavity in a process called cavitation. And so as soon as cavitation occurs, we move from a primary follicle to a secondary follicle. And at the start of our secondary follicle, we still have a primary oocyte. Now the reason why women typically only ovulate one egg is that the follicular cells produce estrogen. So the more follicular cells we have, the more estrogen is being produced, and estrogen inhibits the continued production of follicle stimulating hormone, so that eventually there's enough estrogen and it inhibits follicle stimulating hormone. And then the follicle that's the most advanced continues to develop, and all the other follicles revert back to a primordial follicle and wait for the next month. So in fertility clinics where women don't ovulate or are having trouble getting pregnant, we use high amounts of follicle stimulating hormone to actually make them multiple ovulate. And similar to the physiology of a cat or a dog, in essence. So as we continue over, what's going to happen is the primary oocyte is going to undergo meiosis 1. And typically it's undergone meiosis 1 by the time the follicle matures. So this large follicle down here is a Graafian follicle, or a mature follicle. And Graafian was an early OBGYN who did a lot of work on sexuality in women. So he's got several things named after him. And at this point, the egg looks dinky because the follicle is quite large. At day 14, ovulation is going to occur. At day 13, the day before, we get a peak of a secondary hormone called luteinizing hormone that causes this follicle to greatly increase in size and eventually rupture the surface of the ovary, which is how the egg gets out. So this is an ovulated secondary oocyte, which is the end product of meiosis. And we know it's a secondary oocyte because there's only one polar body, this dark spot. And so what's happened is we've undergone meiosis 1. This retained all the cytoplasm, and the extra nucleus was ejected out of the cell. And this is what's ovulated, a secondary oocyte. Now, if a sperm doesn't contact this secondary oocyte, then no other development occurs, and the secondary oocyte usually vacates the body during the next period, during menstrual flow. So what happens if a sperm contacts an egg, and this is an enormous sperm compared to the egg, so the scale is not the same. So this is like Moby Dick's, the whale sperm. All right. So what we see here is a sperm has contacted the egg, but we only have one polar body. But the nucleus here is getting ready to undergo meiosis 2. And as the nucleus undergoes meiosis 2, we'll get a second polar body. So now we have a mature egg. We have the first polar body, which is the larger polar body. And we have a second polar body, which is the smaller polar body. And we have the sperm nucleus and the egg nucleus coming together in the true point of fertilization, leading to the formation of a single-celled individual that we call a zygote. So let's use this board to review what we were just talking about. And so what we see here is an anaphase where homologous chromosomes are paired up. Here we see anaphase where sister chromatids are being pulled apart. So homologous chromosomes line up in meiosis 1 and are separated. Sister chromatids are separated in meiosis 2. So this is meiosis 1. So the cells above this are all primary cells. These would be secondary cells, the end product of meiosis 1. And then we have meiosis 2 occurring here. So on an egg, what would happen is all of these cells up here would either be in primordial follicles, primary follicles, and the start of a secondary follicle. And as the secondary follicle maturates, the egg undergoes meiosis 1. And instead of forming two identical cells, which is characteristic of sperm like this, what actually happens is the egg actually ejects this polar body, which would be the first polar body. So notice that we still have chromosomes with sister chromatids held together in a centromere, because what we separated over here was homologous pairs of chromosomes. So if this nucleus looked like that and this wasn't there, then we would have a secondary oocyte, and that's what we'd find in a Graafian follicle, and then that's what would be ovulated. And then if a sperm contacts the egg, then the egg quickly undergoes meiosis 2, creates this secondary polar body, and this mature oocyte, where we separated sister chromatids. So in thinking about chromosome number, we started up here with 46. The role of homologous pairs is to result in cells with 23 chromosomes, but we still have duplicated DNA. So meiosis 2 separates the duplicated chromatids from one another to end up with 23 chromatids down here, and then 23 in the sperm that get together and recreate 46 again. So we don't have a good model for spermatogenesis in lab, but we can use microscope slides. So the structure within the testes where sperm is produced is called a seminiferous tubule. So what we're seeing here is cut through seminiferous tubules. They're coiled tubes. So we see different angles because we're cutting through a tube that's been coiled. And when we look at it, then the cells that are on the outside edge are called spermatogonia. And spermatogonia are stem cells that undergo mitosis throughout a man's life to give rise to new cells that can become sperm. We didn't talk about oogonia when we were doing the ovary, because that occurs during embryonic development, so that we have all the primary oocytes formed by the time the baby girl is born. But because women produce 1 egg per month, men have to produce 40 million sperm per ejaculate, then the amount of sperm production is much, much greater. So we have to have these spermatogonia that give rise to cells that become sperm. So the pattern we're going to see is that we have mitosis occurring in our spermatogonia. They give rise to daughter cells that convert to primary spermatocytes. And then again, primary spermatocytes undergo meiosis 1 and form secondary spermatocytes. And then secondary spermatocytes in this area undergo meiosis 2 and give rise to sperm or spermatotids that we would see toward the center here. And so the pattern is in men is we start with a spermatogonia, we undergo mitosis, one of the daughter cells becomes a primary spermatocyte. It undergoes meiosis 1 and forms 2 secondary spermatocytes. And then each secondary spermatocyte undergoes meiosis 2 and forms 2 spermatotids. So we end up going from 1 cell, a primary spermatocyte, to 4 sperm being produced by the same cell. And if everything goes normally, then we have 2 X sperm and 2 Y sperm that are produced. Now what we're going to do is we're going to look at this same process on that black board again as it relates to sperm production.